Okay, good morning to all of you. So, we are starting our next lecture about the division Cyanophyta. So, in syllabus, it is expected we should know about the distinguishing characters, then thallus organization, ultrastructure of heterocyst, and its significance. So, we will go the one by one point. So, in Cyanophyta, particularly, firstly, it is to be called as a cyanobacteria. This terminology generally as a related due to as they are the common known as a blue-green algae. Cyano means blue, and bacteria generally it acknowledges that which are closely related to the prokaryotic bacteria than the eukaryotic algae. Already we are knowing some members are, particularly cyanophyce members are the prokaryotes, and therefore prokaryote that is there is no specific nuclear membrane. In relation to their origin, they are to be supposed 3.5 billion years old and dominated as a biogenic rips. Rip that is the root type, or they generally occur in the groups. So it is to be called as a rip. Then secondly, as we are knowing, there is having about the 2.5 to 750 generally widely spread in this billion from the billion years ago. Then their origin is to be very historical for this one. So generally firstly they are to be called as the cyanobacteria. Then in their distribution, as they are supposed as the microscopic, then in the marine sediments, pelagic zone. Pelagic, that is the open sea is there, it is called as a pelagic. Then freshwater lakes and the soils. In distribution, different places are there. So pools, ponds of fresh water. Common genera are the Anabaena, Rhyolaria, Cytonema, then Gliotrichia, Oscillatoria. We are going to observe the photographs of each one. Then in seawater, Dermocarpus, Trichodesmium, dam soil, Anabaena, Nostoc, Oscillatoria. Hot springs, they are also reported thermal, Crucoccus, Microcystis, Phomedium. Then endophytic, Anabaena, Nostoc, Gleocapsa. Endozoic, in the inside the animal stomach, Oscillatoria, Simoniella. Then cryptophytes, Phomedium. Crypto, that is the on the rock condition inside this rock. Then epithelic, on the epilithic, so like Gleocapsa, Calothrix or Cytonema. Sapropelic, sapro means on the decaying organism, Oscillatoria angusta and the Lingbaia. And in loose lying agarofilous forms, ball like structure, so Toliphothorix, Cytonema. And certain in the with the lichens, Nostoc, Strigonema, Gleocapsa. So in distribution, cyanophyta is very interesting. Then about their importance generally, as these organisms have two photosystems and to produce the organic material which gives the of the oxygen as a byproduct. It is very important to the evolution in at the time of the Earth's oxidizing atmosphere, and therefore as cyanobacteria they shows the characteristics like the different pigments, chlorophyll A, phycobilioproteins, phycoerythrin, phycocyanin, blue-green color due to the phycocyanin, then allophycocyanin, and the storage food is there, glycogen, cyanophycin granules. Cell wall is made up of amino acids and the sugars are there. In importance, we have to consider about they fix the atmospheric nitrogen into usable forms through nitrogen fixation. When they form, they are the unavailable. So we are knowing nitrogen is near about 72% in this one, but it is fixed only by the cyanophyce. As there are the different types of the changes in the wavelengths or the lights are there, so therefore they are to be having the different colors are there. So wavelength, the cyanobacteria appears of the different colors. Good example also the it is the genera having the oscillatoria. Different forms of cyanophyce members are there: unicellular, filamentous, colonial. So unicellular due to the mucilage, and they forms a colony. Secondly, there are the certain trichomes are there. They are called as a trichomes. So mucilaginous sheath is present, and the filament, it is covered by mucilaginous sheath, and it is not too much in the length. Then mucilaginous, therefore it is called as a trichome. This mucilaginous sheath generally, as it protects the from drying and involved in the gliding. So gliding from that is one place to the other. Sheath having the different colors as red, blue, yellow, brown, due to the high salts are there. Then range of vegetative structures in the cyanophyta. So they are generally single or the palmelloid colonies, unicellular, spherical or the oval, colonial forms. They show due to the mucilage. So you just observe here Anastistis. It is singular colonial form. Crucoccus, like this one. Then Cyanococcus. It is like unicellular forms are there. In unicellular Glyocapsa, you see the Glyocapsa, the diagrammatic and the photograph is there. Then there is the Gliotheca. Gliotheca, it is a few cell colonies are there, right? Then Coleosphaerium, so hollow spherical. So hollow means cavity is there. So on right hand side photographs, so you have to. Then Aphanocapsa, hollow spherical. It is called as a palmelloid stage. Microcystis, then Eucapsis. So Eucapsis or Microcystis, they are having the different. Then secondly, palmelloid colonies are there. So Merisopedea is there. In trichomes, oscillatoria is the good example. In live condition, the apex shows the oscillation, and here shown the fragmentation through the harmogonia is there, right? So you just go through it. Then Lingvia is there, filaments, empty sheath, and the trichome is there. So from it the filament gets the break. So vegetative reproduction is there. Then this genus Formidium, you see there is a maximum without content region is there. So it helps for the vegetative reproduction. Then Spirulina, it's showing the helix like structure, spirulina, photograph of the spirulina. Then Arthrospira, so it looks like the Spirogyra, but the nature is the different. Then Rivularia, many mucilaginous are there, branched like structure. Rivularia bilata, which may be seen on the rock, colonies are there. Then Rivularia bilata, it is a magnified structure. Then Gliotrichia is a field-like structure, but it shows many, there are the trichomes are together. Gliotrichia, it is a colony is very specific, star-like appearance is there. Then our Microcoleus is the one. Cyanobacteria, in cyanobacteria we having the this type of the structure is there. Then as we are knowing, there is having the nostoc colony, or the our nostoc balls are there. So nostoc shows the trichome, and each magnified into the, as it shows like structure, heterocyst. Then akinets are there. Akinets are the storing the reservoir material. So same thing in the nostoc is there, right? Then cytonema. Cytonema shows the specific entire mucilaginous vegetative cell and the heterocyst, then decaying cell. So it is a pseudo branching is there in cytonema. You observe here, it shows the pseudo false branching due to the this attachment. Here it is shown. pseudo branching. So both it looks like a branching, or you see the diagrammatic. So this study is generally related with the research of the many scientists are. So false branching. You see, this is a good photograph for the cytonema, false branching or pseudo branching. Then in algae, the cytonema or petalonema is the one genus is there. You see the appearance. Then branching in the this cytonema. Then in branching, false branching and true branching is also there. So outgrowths of filament adjacent to the dead or the specialized cells. And in true branching, outgrowths of the cell that changes their axis of division at 90 degree from the axis of trichome. So this is one good example in the Tolipothrix. This left hand side genus, it is the Tolipothrix. So go through it. Then nextly cyanobacteria members. So this shows the heterocyst is there. So this heterocyst is the character for the cyanophyceae members. Stygonema. You see the right hand photograph, which is generally seen on the limestones are there. Stygonema. Then it shows the some coastal regions are there, seashore areas. So it shows the haplosiphon. So it shows the true branching is there. Then there is the Mastigocladus. It also shows the true branching. And in the relation to reproduction of this one, Cyanophyta, vegetative, asexual and sexual reproduction is completely absent because no reproductive structures they are developed. So therefore this is the one part is there. Then in Harmogones only develop in the filamentous genera, not in the colony. Secondly, in Oscillatoria, you are observing good slide of this. So see the Harmogonia in the trichomes are there. It is not called as a filament, trichome. So this develop without any content. Then in vegetative mode of reproduction, some examples, it is the pseudo Harmogonia is there. It is not true, developing at the apex. Vestiella is the good example. Then it develop the endospores. They are developed by means of the asexual reproduction. Asexual reproduction generally is due to the change in the water character, pH, temperature, then salt content, and therefore there is asexual. So it disturb the sexual reproduction. In asexual, good example for the chemosiphon, exospores are developed. You see, then chemosiphon clavata, it shows the exospores. Akainite formation, thick walled, which functions in the unfavorable condition. Then cyanotoxins in the cyanobacteria are there. So spirulina is the good example for the this one. So it is filamentous. Common in the high pH and major food for the flamingo populations are there. So we are knowing flamingo birds. So it is good food for the these birds. Then neurotoxins generally they block the neuron transmission in the muscles. So it is due to the NABNA. And hepatotoxins inhibit protein phosphatase, cause liver bleeding, and found in the drinking water. So in water pollution, these are the main some genera are responsible. So this is about our introductory part for the cyanophyta. Next lecture we will take on the heterocyst. Okay, so thanks to all.